Okay, so let's go ahead and do day 3 of in-class review. That means I'm going to be starting on the form I just handed you. And it's going to start on the side with the title and, of course, where your name can be found. So I'm just going to tell you, I tried to get a lot more detailed with this. So hopefully, I'm not wasting as much time. So we'll see. Hopefully that all works out to be all great and wonderful and all that stuff. So let's do this thing. I'm going to start in the middle of the top box. That's going to be the formulas. Formula. I'm bad at writing. Doesn't matter. So let's go ahead and separate this out into these two different processes. So we have photosynthesis. Okay, photosynthesis. That includes these things: CO2, water. And I did an interesting thing here. I kind of added in sunlight like this. I didn't include it as like a normal part of the reaction just because, well, it's kind of not normal. It's solar energy. It's odd. Anyway, it's still on kind of the left side of the equation anyway. And then this is going to produce C6H12O6, which is sugar. And of course, it's going to produce as a byproduct oxygen gas. Now, photosynthesis here has this interesting relationship that we find with cell respiration. And I should just go ahead and point out this is aerobic cell respiration. Basically, it's the same stuff. It's the same ingredients. It's just rearranged. And everything on the right-hand side for photosynthesis ends up on the left-hand side for cell respiration. So it needs both sugar and oxygen gas. And then, of course, it produces CO2, produces some water, and it produces energy. This time, it's not so intangible. This time, it makes something called ATP. Do you guys remember ATP? Yeah. Adenosine Triphosphate, which is a really fancy way of basically saying, hey, this molecule, it's got 3 phosphates on it. And those 3 phosphates hold a lot of energy. Now, it actually forms this cyclic relationship with something called ADP, which is adenosine diphosphate. So it has only 2 phosphates attached. Now, if we break off one of those phosphates, we release a bunch of energy, and then we don't lose the molecule. We still have ADP left over. If we ever want to build that back up, we just find another phosphate, plug it on there, and hey, we've stored up some energy that we can reuse whenever we need to. This is really called phosphorylation, but whatever. Like, let's not worry too much about that. I should also point out to you, everything on the left-hand side of one of these equations, they give where they're given a special name. We call them reactants. And everything on the right-hand side are called products. And you guys may have also noticed just below all this stuff, I put a little star there, and it says chemosynthesis. This is another form of energy collection that doesn't use light. So we've talked about these organisms that live at the bottom of the ocean, where it's totally dark. It's perpetually dark. It doesn't change. There's no light that can actually reach that deep. But they live in really cold, dark environments, and they basically use the energy that's created from the ground, geothermal energy. They use that stuff, and they basically survive off of it from whatever the byproducts and molecules are that are there. So that's kind of what that is. How you guys doing? Okay, so let's like hyper fast like hit up what photosynthesis is. And I'm going to try to simplify this as much as I can. First off, where does it occur? Chloroplast. But those are a little bit more complicated than just that, which includes things like chlorophyll. It involves things like grana and stroma, and even, what's the other one I care about? Oh, the thylakoid. Fun words, but we'll walk through it. So, yeah. It looks a lot better on your paper, but I don't care. All right, so we have basically two halves here, okay? We have light dependent, and we have light independent, the two halves of photosynthesis. One side requires light, that's the left side, and then there's the side that doesn't require light, which is the right side. So since it requires light, so one of the main ingredients is, of course, light. That makes sense. And then, of course, the other thing that's brought in there is going to be water. Now, I just want to point out to you, the things we're pointing at, this whole big blob, is the chloroplast. Inside of this, the little stack of discs that you see, the little pancakes, those are called thylakoids. When you get a lot of thylakoids stocked on top of each other, that's called a granum or grana. This is where the first half of photosynthesis takes place, and it really happens in the thylakoid membrane. And basically the goal is hydrogen needs to kind of work through that membrane, and when it does that, it can make a little bit of energy, a little bit of ATP that gets sent over to the other side. Now the oxygen, plants don't care about it. They don't want oxygen, so this immediately gets released as oxygen gas, and I can breathe that in and life is pretty good. Then over here, we have this thing called the Calvin cycle. And the Calvin cycle basically makes sugar. So what happens here is it takes a little energy from the first half, it takes in CO2 from outside, and then from this it produces sugar, C6H12O6. That's a very, very, very oversimplification of what happens there, and eventually some lower forms of energy will be sent back to this process for it to repeat. But now you know where all the water, where all the carbon dioxide come into play, and when and where things are produced. Does that make sense? I would also just like to remind you guys that you're my favorite third period. Okay, let's move on. Cell respiration. I thought there was something else I wanted to say, but I thought that was poignant enough. You know, that really drove the point home. You're my favorite third period, for sure. Well, you guys always fall short on seventh period. That's my planned period. So, aerobic respiration, that means it requires oxygen, right? Aerobic exercise, you're going to breathe pretty heavy. So let's talk about this process. There are a few things that we should know, but first off, where does it occur? The mitochondria. Yep, you're right. And the other thing that I want to point out to you is even part of this is involved outside of the mitochondria. So there are three real parts to this. There's glycolysis, there's the Krebs cycle, and then there's the electron transport chain, which also involves something called oxidative phosphorylation. Anyway, glycolysis happens in the cytoplasm. It doesn't happen in the mitochondria. It happens before the ingredients can get to the mitochondria. So that's going to be found over here, the mitochondria. Anyway, glycolysis, and I'm going to get lazy here, it takes in sugar and it breaks it in half. Okay, so these half molecules, they're called pyruvates. They're going to get fed into the mitochondria now because regular sugar, it's just too big and complex for our body to digest. That's why we even have like insulin, because sugar needs to get broken down to a more simple version. Anyway, this is going to be brought inside of the mitochondria. Now, the mitochondria have these little folds that we like to call cristae, which I'm just going to get lazy and make them small here. But there's a cycle that happens in the space in between this. This is called the matrix, Neo. So the Krebs cycle takes place here. What happens is these chunks of sugar are brought in, and basically what's going to be released, as you can see on your paper, is carbon dioxide gas and a little bit of ATP. I should actually tell you it makes about 2 to 4 ATP. It actually varies on its cycle. And back here for glycolysis, it makes 2 ATP as well. So we're making a little bit, right? Yeah, we might spend a little energy, but we're making a little profit. But it's nothing to really write home about. But when we get to the electron transport chain, some energy is kind of transferred through these processes. And basically all of this is going to happen here in these little membranes. Man, that sounds familiar. It sounds almost like when we talked about it in the chloroplast, right? We said the thylakoid membrane. Well, in this case, the mitochondrial membrane is where this process is taking place. So we've taken the two halves of photosynthesis and we moved them. We just swapped places with them. When this happens, what we do is we bring in oxygen gas, O2, and from this, we break down that oxygen gas. We make some water, and we also make ATP. Now, this ATP is a lot. It's like 34 ATP. It's a big ramp up in terms of production. Now, the reason for this is actually the same reason why photosynthesis makes a little bit of ATP. It's because of this molecule called Atp Synthase, which means if you pass hydrogens through it, you can force phosphates onto ADP. And just by chance, the way cell respiration is set up, it just does this a lot and really, really, really effectively. I should also give you the magic number that this thing basically makes about 38 ATP. I don't know if that number is going to show up on the EOC, but I just want you to be aware. Glycolysis makes 2, the Krebs cycle makes 2 to 4, and of course the electron transport chain, it makes a ton. It makes 34. So total, about 38. It could be more, could be even slightly less, but hey, there you go. So, do you guys have any questions on cell respiration? Notice how I'm trying to be a little bit faster, a little bit snappier? Okay, all right. No questions? Question? No? No? So fermentation. Okay, so fermentation, it involves that process, once again, that we already mentioned, glycolysis, glyco sugar, lysis, breaking sugars. So we basically break sugar and make 2 ATP. Now, this is all what we call anaerobic. And anaerobic is basically saying, hey, no oxygen is needed, no O2. As a matter of fact, most cellular processes that are anaerobic, they'll shut down if oxygen even shows up. That's why like winemakers or like people that make whiskey and stuff like that, they seal it up in like bottles or barrels and stuff like that. If there's ever a leak in there, well, it kind of ruins the batch. So if there's ever like some kind of, I don't know, bad thing that happens in the process, somebody drops a bottle or a barrel and there's a little crack, well, they might have ruined the whole thing, and then it might become vinegar. But anyway, so the main things between these two, the two forms of fermentation we really care about, are going to be lactic acid fermentation and alcohol fermentation. There's a lot more than just this, but these are the only two that we really care about because their products are a little bit different. So remember, we start off with sugar in all of these. We're going to break these things down into two things called pyruvates, or pyruvic acid. And remember, there's 2 of these, and that's going to happen in both processes. But things start to twist. Things start to get a little bit different, because honestly, everything we just drew right there is glycolysis. That's all it is breaking down that sugar. But the interesting thing that happens in lactic acid fermentation is we make something called lactate, which you know is lactic acid, and it makes 2 of them. Now, this is the byproduct that's found in our muscles. So if you ever work out and you're just doing like an isolated exercise, you guys remember my embarrassing 9th grade story where I worked out for the first time and I couldn't move my arms or my chest and I couldn't pick up the remote so I could watch cartoons later that afternoon. Yeah, all in the same day. Great day, right? So all that lactic acid is super painful because, you know, that literally our cells and our muscles are like, hey, I need more oxygen, or I'm going to do some other things to get some energy so I can do all this activity you're asking me to do. So lactic acid is the result. Also, some bacteria use this, or fungi use this, like if you've ever heard of like cheese making or people making yogurt. The thing that adds that tanginess or that sharpness to some cheese, that's because of lactic acid that's found there. And then we have alcohol fermentation. So another interesting thing happens here. Not only do we make 2 alcohol molecules, but we also make a little bit of CO2. So there's an extra byproduct here, which some would argue this is the most profitable biological process on Earth because we use it for all kinds of things. I mentioned alcohols earlier, like wines and and whiskeys and stuff. But also like champagne. If you've ever seen people pop open a bottle of champagne, tons and tons of bubbles come out, right? Because that's a byproduct of that process. It's natural. Those bacteria or fungi are making CO2 as part of their process, and they leave behind the alcohol. And the cool thing is that when the alcohol is made, it kills off those living things because there's just too much alcohol. It kills them. The other thing is in baking we use this because you mix yeast, fungi, with sugar and water and flour, and you basically use water to wake up the yeast eat the sugar, and then after they eat the sugar, they burp and they fart out CO2 gas that gets trapped in the flour, which makes your bread fluffy and kind of nice and wonderful. So alcohol fermentation, it's a big deal. Overall, I just want to point out to you that glycolysis forces, let me phrase this again. Lactic acid and alcohol fermentation force glycolysis to continue. So because they both start off with glycolysis, it's just basically the end products kind of feed back into it and say, hey, we're going to continue doing this. We're going to keep making energy. Okay? Doing all right? Questions? Concerns? No? All right. The cell cycle. I've told myself I'm going to own a bike shop one day, and I'm going to call it the cell cycle because everybody's going to think it's always on sale. Okay. So, interphase. Interphase is basically comprised of 3 parts of the life cycle of a cell, and that's what the cell cycle is. It's the stages of life. There's an early stage, a middle life stage, an end life stage. Anyway, so we've got gap 1, we've got synthesis, and we've got gap 2. Now, I just said gap, but really you should think growth, because what occurs during G1? Growth. You could also say they mature, and it basically operates normal. It operates normally, so it's going to carry out normal cell processes. Then synthesis stage. What do you guys think happens here? Yeah. DNA replication. So DNA replication is pretty important. Thanks. I got it. DNA replication, pretty big deal, and that's when we're making exact copies of the DNA that are already found inside of the cells. And then G2, well, this should not be a surprise because we go through growth. It still matures, and it still operates normally. So the cell, for a majority of its life, is just doing cell things. If you'd like a copy, there's one up there. And then there's a, well, let me kind of draw it for you. I know it's already on your paper. Here we go. Hey, look at that. That's awful. Not much better, but still kind of round. And it kind of looks like a peace sign to a degree, but then we like kind of narrow things down a little bit. Okay, so this is G1, synthesis, G2. These are the 3 parts that make up interphase. Don't forget this. This is all just interphase, the normal life stages of a cell. But there can be an exception. There's a thing back here called gap 0, okay? So gap 0 is basically when cells don't divide. You'll see how it kind of loops within gap 1. It basically stays in that stage. It just kind of hovers there and won't do anything. You have cells like your liver cells or your nervous cells, like your brain cells. Those don't grow and divide. They don't change. We don't want them to change, but maybe your liver cells can be kicked out of this if there's damage to the liver or you basically have surgery or something. But your brain cells and nerve cells, once they're damaged, they kind of stay that way because they don't come out of gap 0. Then we also want to go ahead and define some of these important terms. We've got mitosis, which mitosis phase happens right here, mitotic phase. And mitosis is the division of the nucleus. And then there's cytokinesis. Who knows what cytokinesis is? Yes, ma'am? Yeah, it's the cytoplasm's division. Now, these are different things, even though I know we talked about these parts yesterday. The nucleus needs to divide first, because in the cell cycle for eukaryotic cells, I should point out, they need a very precise process. This DNA, it's really, really, really necessary that it gets done perfectly. Whereas it comes to outside of that, then we're just kind of cutting the cytoplasm in half. That's cytokinesis. And that's what this reddish area that I kind of drew up here was. That's cytokinesis. And through this life cycle of the cell, we do know that it also makes checks. That's what I kind of drew here, were these check boxes. And basically at the end of each of these stages, there are checks that need to occur. Now, if there's a check and there's a mistake, in normal cases, the cell will recognize it and say, hey, let me destroy that thing. Let me try again. We made a boo-boo. But if it somehow makes it through these checkpoints and there are mistakes and they're not noticed, and the cell moves on to go through mitosis and cytokinesis, we've made basically an irregular cell. And more than likely, they're going to reproduce even faster, and those mutations, those problems are going to get worse and worse and worse, which can grow into a mass that we call a tumor. Some tumors are benign, some are malignant. Benign are basically, they don't spread, and they might grow, but that's about it. Malignant can spread, though. And those are the really dangerous cancers that nobody likes to hear about. So that's basically the uncontrolled cell growth. I also want to point out to you, there are these things like CDKs, or really cyclins, and kinases. These are proteins that build up in certain amounts as we go through the cell cycle. And once you get enough of them, it's kind of like a checkpoint and says like, yes, you've earned enough experience. Move on to the next level. And that's what happens as they move through there. As they build these things up, they can connect and form these complexes, which kind of moves off and lets them do their thing. There's even MPFs, mitosis promoting factors, but whatever. So we've got the cell cycle. How you guys doing?
You're good. How are we doing on time?
Pretty solid. Amazing, actually. I don't know what the last thing you said was.
Every day for the rest of the year.
How are we doing on time?
How are we doing on time?
Thank you.
About like the middle of the day.
About.
You'll just know whenever it starts.
I know. I just get that feeling.
Yeah, I get it. You got to stop watching your heart, and it says ask. I understand.
Phones allowed.
So I said stopwatch. Stopwatches are like things other than phones.
Are those the things that you put in a pocket?
Sure, you can put a lot of things in pockets though. So binary fission. Binary fission means we're basically going to split things in half. Now, I'm just going to make a note here and say this is only for prokaryotic cells, prokeys. Eukaryotic cells don't divide this way. So let's get the simple one out of the way first. First off, this is an asexual form of reproduction. Asexual means, do you need a partner or do you do it by yourself? It's by yourself. So these guys don't need a partner, which means even one single prokaryote, like a bacteria, can reproduce and make a lot more bacteria that depend on each other. There's no particular stages. I've kind of written this on your form. There's no particular stages when it comes to binary fission, but the basic process looks like this. We have a cell with a copy of its strand of DNA. Remember, their DNA is really short, like very tiny, so they can reproduce really quick. And what happens is they make a little bit more space, and they go through DNA replication. They basically make 2 copies of that DNA, and it's done in a different capacity than even what we see for eukaryotic cells to a degree. Eventually, what's going to happen is these things are going to start to stretch, and the DNAs are starting to move to opposite sides of their cells, and then eventually, it's going to split in half and make 2 new cells. That's binary fission. It's simple. That's one of the reasons, again, that I make fun of prokaryotes, because they just kind of rip themselves in half and hope for the best. That's all there is to it. Now, when it comes to us, though, we use that really precise process. But do you guys have any questions on the binary fission process?
They're trying to demonstrate.
They what?
They're trying their best.
They're trying, and honestly, it's very effective. Like, dang, dude, like they clone themselves in like 20 minutes. That's impressive, and that's a whole new individual living thing. It takes us 9 months to pull that one off. You know what I mean? So good on them, but it's also not like they're very complex. So let's get through mitosis.
It's not their fault.
No. If anything, it's like the OG version of doing everything. So Once again, mitosis is an asexual form of reproduction, so it doesn't need a partner. So our cells do this whenever we grow or we repair ourselves. I should also point out to you, we start off with one diploid cell, and from this, we're going to make 2 diploid cells. We'll define diploid and haploid later on, but I just want you to remember, diploid is used in mitosis. Also, I've kind of written the stages up here. You might remember my really gross story or analogy of me peeing on one of your bathroom mats accidentally. And PMATC, I peed on your mat. See? So you'll probably remember that, and those will be the stages of basically the cell cycle, but overall the stages of mitosis. So let's go ahead and draw one of these things really quick for you, even though it's already on your paper, I know. What this is it's a chromosome, right? Now the chromosome has 2 halves. Each half is called a chromatid. And then there's this middle portion that's kind of connecting it all together. That's called the centromere. And of course we have the short arm, which is called the P arm, and the long arm, which is called the Q arm. What do they stand for? I don't remember, but it doesn't matter. The point is, these are chromosomes. These form in mitosis because it helps organize information in a more reliable way, and in a lot of cases, these will just be ripped in half by the chromatids, so the 2 lengthwise halves, which are really single chromosomes, but hey, who's keeping count here? So let's go ahead and talk about prophase, metaphase, anaphase, telophase, and then cytokinesis. What happens during prophase? Anybody remember? Yes, sir?
Those little things that rip them apart, bro. The spindle fibers.
Yeah, that's true, but something's got to happen even before then.
We gotta duplicate the information.
Well, that happened during synthesis stage, so that was before mitosis, but good try. Chromatin condenses into chromosomes, and also the nucleus disappears. I probably misspelled disappears, but I don't care. So we've got metaphase. What is going to happen during metaphase? Well, those spindle fibers grow. And I guess they're kind of always growing. But what else really happens?
The chromosomes align in the center.
The chromosomes align in the center. Yeah, it's like roll call and boot camp. That's exactly what it is. They line up in the middle. They're equally spaced, and they're found in a nice single file line. In this case, the spindle fibers are now attaching to the centromeres of each of the chromosomes, and that'll happen on both sides of them, because what happens next is that we start to split the chromatids. Yeah, my analogy I used was like the giant spiders that rip in half the children, right? The spindle fibers are like giant long spider legs. The children are all terrified and they're trying to equally get away from the spiders, but the spiders inevitably find them and rip them in half lengthwise. So anaphase splits the chromatids and it moves. I know it is. Moves to opposite sides of the cell. Then telophase, which if you want to call it telophase, that's fine, but nobody's going to hear you say anything when you take your test, so it's all good. Anybody know what happens during telophase? No? No explosions. What do you think?
The nucleus recondenses.
So the nucleus reforms. And it's around the 2 new sources of nuclear material. And also, the organelles have moved too. Meaning all the organelles that were found inside of the cell, well, they've also spread apart to hopefully equal amounts on both sides of these cells. Then we've already talked about it, but cytokinesis is when we actually have the new cells being created. We have 2 brand new cells, nice and healthy and ready to live their lives and help you continue yours. So you guys do realize on your papers there you have like 5 drawings. I am not going to draw what's inside those. That's crazy. But I gave them to you in a goofy order just so you can kind of identify them here and make your life a little bit more, I guess, challenging. The first things that happen, of course, are interphase, I, PMAT, C. So interphase is going to be happening on the center one here. So I'm going to put an I on this one. You probably want to label it as well as either I or interphase, because that's what the cell normally looks like. But then what happens is prophase. Prophase, those chromosomes condense, so those X shapes start showing up. So it's going to be that second picture that you see. And then what's going to happen is that they line up. That's the metaphase plate. So we're going to label this one on the far right as metaphase. Then anaphase. Anaphase. The chromatids are being separated. So anaphase will be on the far left. And of course, telophase will be our last and final remaining box. Try to keep in mind, cytokinesis just cuts the cytoplasm afterwards. So, do you guys have any questions on mitosis? Yes, sir.
Yeah, why do you get the pictures like this? Why do you scramble them up?
Because I had my TA draw those things for me, and I showed her a picture of what I wanted her to do, but I forgot to tell her, please put them in the proper order. So, it was my fault. But that's fine. I can fix it pretty easy by doing this, right? Groovy. So, everybody feels good? Okay. Let's do this thing. Meiosis. Meiosis, we're going to keep this pretty simple. Meiosis basically works out. How do I want to do this? Well, it's no longer a asexual form of reproduction. We're going to be using this for only sexual reproduction. So let's define what diploid and haploid are. Diploid and haploid. Diploid means it has a full set of chromosomes.
Haploid is half.
Yeah. Okay. So diploid, full set, haploid, half set. In meiosis, we go from one diploid cell, and we're going to make 4 haploid cells. Not 2, 4. And we're also going to make sure each and every one of those is unique. They're very, very simplistically stated unique cells. There's differences that are found in every single one of them. Now, sorry, I'm trying to look through everything in the world now. But anyway, so let's go ahead and look at that really fast. We have one cell that's going to divide into 2. I'm not going to draw the centers of these cells. Sorry, I'm lazy. But then we're going to make 4 cells. Now, the reason I'm showing this to you is because there's 2 stages of division. It divides twice. We call this meiosis 1, and we call this second part meiosis 2. Yes, we use Roman numerals. Why? I'm not sure. But when I wrote this information for you, I wrote I, PMAT, C, PMAT, C. Oh. And there's a 1 below these first ones, and there's a 2 below these second ones. I know it looks like a bunch of commas, but I was really trying to write 1s and 2s there. So if you need to fix that, go ahead, because this represents meiosis 1 and meiosis 2. So we know we see 2 different stages of division. I should also just point out to you, there are some very complex processes that also happen within this. So I'm going to try to draw this crossing over. Hopefully you guys will remember this. So let's say I got chromosomes that are similar to each other, homologous chromosomes. They hug each other really, really close, and they're the same size. And you'll see when they hug each other really, really close, some information will be exchanged between the 2 of them, right? Do you guys remember my analogy of the ant at Thanksgiving or whatever it was, and she hugs you and she leaves behind some animal hair on me, and I smell like old lady, and maybe there's lipstick on my cheeks because she kissed me? Yeah, that's going to be like my analogy of crossing over. So what is crossing over? It's when homologous chromosomes share genes with each other. This increases genetic diversity, which is key to meiosis. We don't want to make exact copies. We want to make half copies that are still different from each other. So every single sperm that's produced or every single egg that's produced has a chance at having something different about them, which gives equal opportunity for genes to become more popular or less popular, hopefully based on the traits that they actually have later on. So then there's independent assortment. Now, independent assortment is interesting because basically, I will draw it for you, but let's go ahead and define it first. Independent assortment is going to be, the, we'll go ahead and say the random movement of chromosomes that doesn't depend on where other chromosomes go. So we've kind of talked about this before. That's a small x and a big X. And let's get a blue color. A big X and a small x, a big X and a small x. If this is within a cell, we know we're going to split these, right? And they're going to end up in two new cells. Now, there's a chance, there's actually equal opportunity for the big blue one to end up on the left-hand side and the little blue one to end up on the right-hand side. The same opportunity that there would be for the big blue one and the small blue one to end up on the same side. It doesn't matter what the red one does as long as the same size chromosomes separate. They can separate whatever direction they want to. Does that make sense? Creates variety in where those chromosomes can move. So in this case, you know, of course, the big blue, I gotta change colors. The big red one could end up over here and the small red one would end up over here. But we could have just the same possibilities that it's all red on the left and all blue on the right. They act independently of each other. The chromosomes can move wherever they want. Okay? This is why when we build Punnett squares, by the way, remember Punnett squares? That's why we always put both letters and we cross them with all the possibilities, because it's possible that they can all show up wherever they want to. That's why it gets more complex if we add more chromosomes, more alleles that are separated. Doing good? Okay. Let me find a thing so I'm not wasting your time. So we just have one final table. We can do this. So I'm going to do mitosis. I'm going to do meiosis. This is just like a fact sheet. So here we go. When it comes to the type of information it is, well, mitosis is going to be, of course, asexual. And meiosis is going to be sexual. It's used for making sex cells, okay? When it comes to the parent chromosome number, so we'll just say parent, okay? The number is going to be 2n, which 2n always means diploid, okay? And then in meiosis, we still start off, the parent is still 2n. It's still a diploid cell. Next up, we have the daughter cells, or how many daughter cells are going to be made, right? So I'll just put daughters. The daughter cells that are produced, of course, are in asexual reproduction, and mitosis are going to still make diploid cells. Now, we'll have 2 of them, but each of them will be diploids. And in sexual selection, we'll have N, which now, in this case, means haploid. N just means chromosome number, so we're getting half of what would be there. 2 full sets of information is a diploid, half that is a haploid. So we've got our daughter cells. When it comes to the number of divisions, try to remember, there's only 1 stage of division when it comes to mitosis, but there are 2 in meiosis. When it goes to the number of cells that are produced, well, of course, the byproduct is going to be 2 cells in mitosis, but it's going to be 4 cells in meiosis. And then replication, DNA replication. When does that take place? It'll take place in interphase. And I should just point out during S phase. Remember how we pointed that out? Because that's a regular part of the cell cycle. That will also occur during meiosis. It'll still take place during interphase. But we're going to make a note here and say before, and only once, It'll happen before meiosis 1. So it only happens once. We don't go to interphase in between. We don't do that again. We don't go through DNA replication again. There's 2 stages of division, but only 1 stage of copying. So do you guys have any questions on that? I don't know how I did it, but I finished in the class period.